Before we leave this slide, I cannot impress on you enough. Anaerobic respiration and fermentation are not the same thing. They are not the same thing. Anaerobic respiration and fermentation are both anaerobic processes, meaning neither process uses molecular oxygen. However, anaerobic respiration requires the TCA cycle, right? And also the electron transport chain. Running of the TCA cycle and ETC are not present in the fermentation pathway. Fermentation is a simple process which consists of glycolysis and then fermentation. As far as what process will run in what organism or what cell and tissue type of what organism, it has to do with what genes are present and what genes are turned on or off in those different cells and tissues. So there's two general approaches to making ATP on the molecular level. And again, the ATP is the adenosine triphosphate, preferred energy currency of all cells. So the number one way of making ATP is oxidative phosphorylation. The approach to oxidative phosphorylation requires the use of ATP synthase. We're referring to this as oxidative because of the redox reactions in the electron transport chain. And we're providing hydrogens and electrons to the electron transport chain by these reduced cofactors: nicotinamide adenine dinucleotide hydride and flavin adenine dinucleotide dihydride. The electron transport chain is going to oxidize these reducing equivalents to generate a hydrogen ion gradient. Hydrogen ion gradient is what provides the proton motive force, which is often abbreviated as PMF, proton motive force. And that proton motive force is derived from the hydrogen ion gradient in the mitochondrial intermembrane space. So this is like a battery. It's like a hydrogen battery where there's a lot of potential energy stored in the form of this high hydrogen concentration within the intermembrane space of the mitochondria. So there's your outer membrane, there's your inner membrane, and this is your intermembrane space where we have lots and lots and lots of hydrogen ions. These hydrogen ions provide for us the potential energy which we call the proton motive force. The proton motive force can be harnessed in order to do some cellular work. Within this inner membrane of the mitochondria, so there's your inner membrane of the mitochondria, the intermembrane space is at the top here, and the matrix is shown at the bottom. We have the hydrogen ion gradient very high in the intermembrane space, and we have hydrogen ion concentration very low in the matrix space. And within the inner membrane of the mitochondria, we have protein complexes through which the hydrogen ions can flow, and we use that driving force to do some cellular work. For example, one of these protein channel doorways is the base of the ATP synthase, and we harness the power of relieving that hydrogen ion gradient to power the formation of ATP. While we're on the topic of oxidative phosphorylation and the concept of the electron transport chain machinery forming a hydrogen ion gradient, we can look at some evolutionary conservation here. So let's look at this idea of the location of the electron transport chain machinery, or the ETC protein complexes. So if we look at gram-positive bacteria, Gram-positive bacteria have only one membrane, a plasma membrane, and that electron transport chain is embedded within that cell membrane or plasma membrane. If we have gram-negative bacteria, that electron transport chain machinery is going to be embedded within the inner membrane of the gram-negative bacteria, not in the outer membrane. In our gram-negative bacteria, the hydrogen ion gradient is going to build within the intermembrane space. If we look at gram-positive cells, the hydrogen ion gradient gathers just outside of the cell membrane. And then if we look at the equivalent for our mitochondria, right, we have the hydrogen ion gradient in the intermembrane space. And so if you think about the endosymbiotic theory, the location and function of the electron transport chain supports endosymbiotic theory. The endosymbiotic theory provides a hypothesis for the origin of organelles in eukaryotic cells. The second molecular approach to making ATP is a minor way, and by the way, this oxidative phosphorylation is a major way. This minor way of forming ATP is really kind of like it's incidental. It is, an afterthought. And so in substrate level phosphorylation, which is the way that ATP is formed in glycolysis in the TCA cycle, or there are some other reactions as well, the point of that chemical reaction is to transform the metabolic intermediate. It is not to make ATP. So the main purpose of that reaction is to transform a metabolite from one molecule to another. Oh, and by the way, in the process, accidentally, some ATP was made. So these are incidental ways or minor ways of making ATP. If we look at these major catabolic pathways for burning, and really elegantly, the elegant way of expressing burning, you say oxidizing, of oxidizing glucose. We looked at these on the previous image, which was this image, but now it's written out for you again here. So when oxygen is present, we can run aerobic respiration, which is a high energy yield pathway, meaning we get out a lot of ATP. And then we have, when oxygen is not present, the running of anaerobic respiration, which is the same as aerobic respiration, right? Because respiration, but we're going to be using an inorganic molecule, which is not molecular oxygen, as the final electron acceptor. Oh, by the way, another anaerobic process, but it's not the same as anaerobic respiration. We have fermentation. This is a great table, also another table from a microbiology text here. So you can see these different energy producing processes, which I don't, oh goodness gracious, I need to come back to life as a textbook figure editor. So we have, I should say, ATP producing processes, right? Processes for generation of ATP. And these are the general approaches, whether or not we have the presence of oxygen or not, and lists out for you the final hydrogen acceptor. I don't like this terminology either. It says an organic molecule here, pyruvate usually. And anyway, then the type of phosphorylation used to generate ATP. Substrate level and oxidative. Substrate level is occurring, of course, in glycolysis and TCA here. Substrate level phosphorylation to form ATP is happening only in glycolysis, in fermentation. And right, oxidative formation of ATP is always referring to ETC, which makes a hydrogen ion gradient, which runs ATP synthase. ATP synthase is the complex which is going to phosphorylate ADP and form ATP. And there's this warning once again. So be really careful about what you look at on the internet. There's a lot of places on the internet that says fermentation is anaerobic respiration. It absolutely is not. Here's the structure of ATP. Please learn the structure of ATP, as in please memorize. I can't test you to make sure that you've memorized the structure, but you absolutely should. It will help you later on in your studies. If we look at this structure of ATP in detail, I'll cross this out so you can see it. This is your adenine ring. It is a purine. It is a nitrogenous base. It is a nitrogenous base as you would find in DNA or RNA. If we look at this sugar, this is ribose. This is a 5-carbon sugar, and it is shown to you as the full ribose. If we number the carbons here in ribose, carbon 1 is where we have attachment of the adenine ring, carbon 2, carbon 3, carbon 4, and here is carbon 5. If you think about DNA and RNA structure, you always think about 5' phosphate and 3' OH. Every strand of DNA and RNA has a 5' phosphate, so 5 phosphate, and a 3' OH. Get this in your mind, and it will help you always to remember that we have the phosphate attached to the carbon 5 of ribose, and then of course the nucleotide base would, I should say, the nitrogen containing base is going to be attached to at carbon 1, and here's your 3' OH. There's always going to be a free 3' end unless we're using chain terminating, like dideoxynucleotides, or you should think like DD-NTPs that we use in sequencing reactions. So they're chain terminators. If you look here, we've got this 2' OH. That 2' OH is absent in DNTPs, right? So let's say ATP that's incorporated into DNA, right? So we have deoxyribo, D, nucleic acid, DNA, deoxyribonucleic acid. So that's the deal. If you look at the different terminology here, it does matter. Adenosine triphosphate is the entire thing that you see here. That's the whole thing. Adenosine is your adenine plus ribose. Okay, so adenosine is adenine, which is just the purine, plus the ribose is adenosine. So I think that the S, I think that the sugar is there as well. So adenine plus the sugar is adenosine. And then we have, this portion is adenosine, triphosphate. There's 3 phosphates. Now we can break off those phosphates 1 or 2 at a time. And depending on which of these bonds we break, we're going to be releasing a different amount of energy. So these phosphates are alpha, beta, and gamma phosphates. To follow again the Greek alphabet. More about that coming up. If we cleave off the gamma phosphate, we will then have formed inorganic phosphate, or PI, and ADP, adenosine diphosphate. If we cleave at the beta phosphate and we release, so this was gamma, if we release gamma and beta, then we have released inorganic pyrophosphate. Pyro means fire, right? So this is a very high energy molecule. And it doesn't accumulate appreciably within the cell. Usually, as soon as we form this inorganic pyrophosphate, it is converted to two inorganic phosphates by an enzyme called pyrophosphatase. Okay, so if we cleave here, we're going to then be releasing inorganic pyrophosphate and will yield AMP, adenosine monophosphate. Okay, some more terminology here on these phosphate groups before we go on. We have a situation where there is the rest of the molecule, and then we have this phosphoester linkage. We form a functional group which is known as a phosphoryl group. So the boxed part is the phosphoryl group. If you look at this context, we have formed a mixed anhydride. If you look in this box, that's the boxed part is a mixed anhydride. And this portion here, you see this carbon here? It's implied that this R is a carbon with something else attached to it. So this, if we're looking here at this carbon, we would say that this is an acyl phosphate. And it's an acyl, right? We're assuming that there's a carbon here. So two carbons, acet, acyl. So acyl phosphate, because we have a phosphate attached. So that's an acyl phosphate. And then if we look at this, and we just change it a little bit, if you look in the boxed area here, that's a phosphoanhydride. So why go through this? Because that goes through the difference between phosphate and phosphoryl. Phosphoryl is a functional group. Phosphate is an anion. So there's three phosphates here: the alpha phosphate, the beta phosphate, the gamma phosphate, right? Alpha, beta, gamma. And there's three positions here where we can have nucleophilic attack by an electron-rich atom like oxygen, right? Oxygen's a good nucleophile. Guess what this phosphate is? See if you remember from organic chemistry. This phosphate is an electrophile, right? He's an electrophile, a little bit positive, right? This delta plus is partial positive charge. Why? Because this oxygen that, or I should say these oxygens that are surrounding this phosphate are electronegative. And so these guys, this one here is partial negative charge. This one here is partial negative charge. Of course, this is full negative charge and full negative charge. So this phosphate is really electron poor, and that makes it hungry for electrons. So this is an electrophile. This oxygen here is a nucleophile, which is, right, again, electron rich. So, hey, I need some electrons. Hey, I got some electrons. I'll give you some. And so what happens is we can have nucleophilic attack. If we have nucleophilic attack at the gamma phosphate, then we can perform a reaction, right? That is a phosphoryl transfer, because we're transferring a phosphoryl group to what was an alcohol. And then we yield ADP, right? And so that would be the cleavage point there. If we have nucleophilic attack at the beta phosphate, then we have a reaction called pyrophosphoryl transfer, where here is our pyrophosphoryl group, and we will then be releasing AMP as our leaving group. If we have nucleophilic attack at the alpha carbon, we will be performing a reaction called adenylyl transfer. And in this case, we are going to have the PPI act as the leaving group. And we have transferred an adenylyl group, which is this, to our nucleophile. The location of the cleavage is important because different amounts of energy will be released depending on what bond is cleaved. Is it this bond, this bond, this bond, right? So it matters which bond is cleaved. And you can see here the table that you've already seen in your homework. To a large extent, the amount of energy released by the hydrolysis of ATP is going to depend on not just what bond is broken, but also the relative concentrations of the substrates and products at the start of the reaction. Why should we care about this? Because this concept, right, how much energy is released, has implications for actual delta G, the actual release of energy upon the cleavage of ATP. So it's going to depend on what bond and also what concentration of reactants and products are present right at the start of the reaction. So actual delta G or delta G sub P, which is the phosphorylation potential, and more about that soon. If we look here at ATP, there are several factors that contribute to the large negative delta G upon cleavage of ATP, and we're going to go through them one at a time. Number one, right here, the cleavage relieves electropropulsive strain. And so if you see this, see all these negative charges on the oxygens and the phosphate groups, a lot of negative charge is going to cause a lot of electrostatic repulsion. And so this oxygen is crowded by this one and this one, and this one's crowded by this one and this one, and they're all trying to push away from one another, but they're tied together by these bonds. So if we were able to cut one of these bonds, that would be a wonderful release of this high tension, high energy system that we have due to all of this negative charge being constrained in one location. So cleavage relieves that electropropulsive strain on the oxygens of the phosphate groups. Number two, right, another factor contributing to a large negative delta G is that we can have resonance stabilization of inorganic phosphate. So here's PI, and you can see this resonance stabilization. This is not possible when we have the phosphate group constrained by a phosphoester linkage to an adjacent phosphate. So it forms a situation of a phosphoanhydride. That resonance of the electrons around the phosphate and through the phosphate and oxygen bonds is not possible when we have phosphates connected one to another. So if we release inorganic phosphate, we have resonance, right, of the electrons in these phosphate and oxygen bonds. Resonance stabilization means that we have a low energy state, right? So stabilization, lower energy state. And so this is going to further drive, right, we're talking about delta G between, here we have ATP as a high energy molecule, and down here I'm going to put ADP plus PI. is a lower energy molecule, right? So why is this high energy? Because of electro repulsive strain, right, of the oxygens and the phosphate groups. And then down here we have Pi as a low energy molecule. Why? Because of resonance stabilization. Third point here, ionization of the product contributes to the stabilization. And as far as this ionization of the product, we can have greater solubility and hydrogen bonding with water. And if we look at that on the next slide, right here you can see we have ionization. So here it is protonated, here it is deprotonated, here it is ionized, deprotonated, same thing. And so we have another full negative charge, and then this allows for more interaction with water. So it's more stable. The more bonds you can make with solvent, the more stable. We are aqueous or water-based systems, and so the greater number of hydrogen bonds, the greater the solubility and the greater the stability. If you look at ATP and magnesium, they kind of go hand in hand. Pretty much any reaction that requires ATP, you also need to provide magnesium to that enzyme, and that's because magnesium shields negative charge on the phosphate groups. So where you see ATP, there is usually magnesium, and the same goes for ADP. So there's usually a magnesium there. So that means that the net overall charge on ATP is going to be only 2 minus in the presence of magnesium instead of 4 minus in the absence. And then for ADP, in the presence of magnesium, we have a 1 minus charge instead of a 3 minus charge in the absence of magnesium.